Ba, Good evening to all participants. We will be starting in another 10 minutes. Good evening to all present here. We will be starting our day three of nurturing future.
Ashweta, your voice is not coming.
Sorry, there was a technical issue from my end. I welcome once again all the participants to our day three webinar of nurturing future biotech industry professionals. Am I audible now?
Yes, yes, please
Sorry,
continue.
there was a technical issue from my end. I welcome. I am Ashweta Iyer, your Master of Ceremony for this 1 week long webinar. I will be doing the opening and introducing our first speaker, Mr. Justin Moses, and this will be carried forwarded by my colleague, Ms. Riddhi Upadhyay, for the introduction of the second speaker and the closing. Um, with here, with us here is present Mr. Justin Moses. He's the Senior Executive at Biocon Biologics India Limited, Bangalore, specializing in mammalian downstream biosimilar production since June 2018. His expertise includes operating various chromatography systems such as SPAL, ACTA, and Millipore, as well as tangential flow filtration systems. Justin is skilled in handling sartorius filling stations, freeze-thaw systems, biosealers, and integrity testing equipments. He oversees downstream processing, including centrifugation, chromatography column operation, viral removal processes, and excipient formulation. Mr. Justin also manages cleaning and sterilization processes, packing chromatography columns, and contributes to novel product development and validation batches, showcasing his comprehensive proficiency in bioprocessing and biosimilar production. We welcome you, sir.
Hello, can you hear me?
Yes, sir.
Uh, this thing, I can't share my this thing, PPT. Uh,
Wait,
can
sir,
I say?
I'll do change.
Yes, sir.
Sir, now can you try?
No, 1 minute. No, I'm not able to. Can I send it to somebody?
Yes, sir, you can send it to like me. Riddhi, you can WhatsApp me. I'll show it here.
Sure.
Just a minute, sir. It's downloading. Sir, the slide is visible?
Yes, yes. Hi everyone, my name is Justin. Uh, is it audible?
Yes, sir. You can
Yes,
start.
sir.
Hmm. Hi, my name is Justin. I'm working in Biocon Biologics Limited as a senior executive in the Department of Mammalian Downstream. I work with a production of monoclonal antibodies. So these monoclonal antibodies are used in the treatment of cancer patients. Like these drugs are being produced. Um, can you change it to next slide, please? So these medicines are used for the treatment of cancer patients, and we use these medicines for improving the immune system of the body. Uh, so these monoclonal antibodies, they target the um, uh, particular protein to fight the uh, this thing cancer cells. So I work in downstream part. So the production of this medicine has been splitted into three sections in our department. So the first section, it goes as inoculum, that you would be worked in laboratories. So it is based on Petri plate from uh, first cell, master cell to shaker flask. So it is being cultivated from Petri plate to 500 ml shaker flask. So this is being grown with the help of media, and these are uh, uh, let allowed to grow from 2.5 500 ml to 2500 liter. So these growth will be done in the upstream production area. So that is the second stage after inoculum. So inoculation is the first department, and this thing upstream production is the second department. So they work uh, in the area for the growth of the media and our proteins, um, from a small quantity to a larger quantity. So this growth of this media, which you would have studied as log phase and lag phase. So the growth of the cells will be taking a particular time of 1 month. So the upstream process will be taking 1 month. So it will be growing, the production of this cells will be uh, cultivated from this 500 ml to 2 liter and 500 liter, and it will go on to 1 liter and 2005 liter. So that is the basic of which how we are getting this process. So mainly what we are working in is downstream processing. So in downstream processing, we don't require live cells. We just require only the particular protein. So the particular protein is extracellular protein. So we have to take the particular protein. So it will be separated, captured, and purified uh, to the final stage where we get the uh, desired protein. So these proteins will be given to the patients for the treatment. So how we are separating this protein from the media is, there are five sets of main processes which are called as separation and capture, purification techniques, microfiltration, viral filtration, and diafiltration. So this, can you go on to the next slide? So in this separation and capture, what mainly we are doing is, it is the centrifuge which you are working. So you would have seen a lab scale. So in college, in lab scale, it is 50 ml falcon in which we are separating the supernatant from pellet. But how we are separating these processes in large scale. Can you go on to next slide once? Uh, if you see this right side top image, this is called as a continuous centrifuge. In this, product which is coming from the upstream, it will be continuously flowing in this centrifuge, which will separate the supernatant from the pellet. So the pellet will be discarded immediately through the centrifuge itself, and the supernatant, it will be collected to the harvest storage tank, the one which you can see in the top right, top left corner. So in these tanks, our product will be collected. So after separating the pellet and the supernatant, our product will be collected in this storage tank. So this product won't uh, uh, not only it will have our protein, but also the host cell and uh, other media particles and debris present in this uh, 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 centrate. So this will be separated using protein A chromatography techniques. So chromatography techniques are used for purifying, separating the particular protein alone. So in affinity process, which is also called as protein A chromatography. So which in that we will have a chromatograph. Can you change it to next slide? So these are called as chromatographic columns. So our product, it will go inside this column, and our protein, it will bind to the resins, which are the white settled you can see the white settled bed inside this column. So along with that, our protein, it will be attached inside that. So once that is attached, apart from the protein, which are all the unwanted medias and other particles, host cell DNA, which are leachables, which are present in that product, will be washed out with the buffers which we continuously give it in that. Once all those are washed out, we will give a particular buffer to take out the protein from the resin, and those will be treated to the next stages, which are anionic exchange chromatography, in which our product will be positive charged and the resin will be positive charged. So once the resin, which is that white substance present inside the column, and the product is are not attaching together, our particular protein, it will be eluted outside uh, without being attached. So we will understand through the systems that our protein is being eluted outside, and we will collect it. So these techniques are being followed continuously after each uh, stages. So after each particular stages, like affinity, anionic exchange chromatography, then cationic exchange chromatography, the purity of the product will increase exponentially, and our yield will we have to keep up with the yield. So yield is the one where our protein, it should not get lost during the process. So the next step which we are doing will be microfiltration. So microfiltration, why we are using microfiltration is to, when we are using these continuous techniques, it will be treated one after another. So during this process, we might have a possibility of getting some contamination or some foreign particles to be present in our products. So for removing these products, what we are doing is microfiltration. This microfiltration won't allow any particles to pass it through the filters once the product is passed through the filters. So another process which we are doing is viral filtration. So in our product, we should not have any viral particles present, but we are not doing any viral testing. So, but as per US distinct uh, government rules and partners rules, we have a set of regulation. So there are regulatory bodies which give us a set of regulation which to be followed. So as per these regulations, we should have at least two viral inactivation and viral filtration process. So these process will help our product to be free of virus present in the product. And there are another final process which is called as tangential flow filtration. So this tangential flow filtration is to concentrate the product. So in our product, we will have 2 mg of protein or 5 mg of protein before these chromatography stages. After these chromatography stages, once we are about to start tangential flow filtration, our main object is to uh, uh, our main object is to concentrate the product from 2 mg to 30 mg. So this 30 mg, why we are targeting is this 30 mg will be filled in a vial. So this particular 30 mg will be given to the patients. So uh, uh, this by uh, you can see in this tank. In this tank, the product will be present, and the bottom right corner, you can see a section which is called as cassettes. So these cassettes won't allow our protein to pass outside the uh, cassette. So it will keep our protein alone inside this tank. So it will concentrate the product where the volume will be around 500 to 600. And once during 500 to 600, our protein will be around 2 milligram. So once this is done, our process is completed, our quantity will be around 50 liters, where this protein will be around 25 mg. So we have to reduce this protein to 25 mg per ml, so that when we are filling it in this vial, it will be 1 ml equal to 25 mg. So for that process, we are using this technique. In this technique, there is another process which is called as diafiltration. So diafiltration you would have studied in lab scale. It is to exchange of buffer. So before these stages, we will use buffers in the processes. So after this, we will have a particular buffer which will be with the which will be with our protein. So our protein won't get denatured after a particular time. So once this process are completed, we will fill it in through the filling station and keep it in deep freezer, which is minus 60 degree. So we will freeze it in minus 60 degree, and we will store it in minus 20 degree deep freezers. So these buffers and these excipients will help the protein to be in a stable state, and it won't allow it to get denatured or break denatured. So for that, it is being uh, it is being mixed with the protein. So this the whole time will be around 2 years. So for around 2 years, we can store it in minus 20 degree. Can you change it to the next? Next one. And one more. For example, the uh, uh, there are I've given example trastuzumab and rituximab. So in all the monoclonal antibodies, the product name will be uh, completed with maps. So those are called monoclonal antibodies. So as of now, we are uh, working with around 5 6 maps. So each maps will treat particular diseases, to fight against particular diseases. For example, there are 2 maps which are mentioned here. Those are trastuzumab and rituximab. So this trastuzumab, it treats breast cancer, and there are rituximab, which helps in treatment of uh, colorectal cancer. So there are still more maps which are particular for each stages of cancer and types of treatments. Can you change to next slide? Thank you. If you have any doubts, you can.
Thank you so much, sir. So, from one of the participants, Devishri, she has asked why microfiltration is important.
So why microfiltration is important is, see, from this first stage till last stage, there are 8 separate unit operations. So everything won't be done in, for example, you would have studied in labs that in lab and all, for it will be class A, class B, class C, and class D areas. So these areas, why it is segregated is, in class A, there won't be any other particles present. So that's why it has been classified as that one. So this product and these medicines which we are producing will be produced in a class C area. So once we are producing in class C area, uh, uh, the stages like affinity and anion exchange and cation exchanges will be done in class C area. So when we are doing in class C areas, there might be the range of that areas control will be 100 100 micron, uh, uh, in that it can be present in that area. So this process will be continuously changing from one affinity. Once affinity is completed and it is going to next anion exchange, it will be changed from one bag to another, and there might be open operations. So when we are opening up like bags, for all the bags which we are using to collect the medicine, it will be single usage bags. So once the product is getting contacted to the environment, it will be affected for the growth of other particles, any other particles which might present in that area. So those particles, it should not present in our product. So once completion of every process, for example affinity, once affinity is completed, we will treat it with microfiltration. So microfiltration will remove all the particles, even if there are any chances of these microparticles to be present at the end of affinity stage. If there is any a possibility of microbes inside the product after the affinity stages, uh, there will be 1 day time or 1.5 days time for the continuation to the next process. So in this period of 1.5 days, this microbes which is present at the end of affinity stage, it will grow uh, more in quantity before the stage of uh, AX stage. So uh, it will impact our products purity, and it we can't say if microbes any other contamination is present inside a product. We can't say, we can't give it to our patients. So as per guidelines, after each completion of process, we have to treat it with microfiltration, so that at the end of uh, TFF and formulation, there should not be any particles present. Uh, for, any particles present in our product. So, to check microbes present after each, if we are starting a process and if we are completing a process, we will give a sample to check if there are microbes present in the uh, present in our product. So those samples will be given to microbiology team. So they will be treating into a plate, and for us, for example, for us, limit is like 10, not less than 10, not more than 10 colony forming units. So it should not be more than 10 colony forming units. So it will be tested after each process. So that is the reason why we are using microfiltration. Hello.
Thank you, sir. Another participant has asked, please elaborate on the maps used for cancer treatment.
Mm, cancer treatment. I can't give any particular names, but I will give, like, for example, two names which are widely used in markets. Those are trastuzumab. Uh, Trastuzumab is used only for breast cancer, and there's bevacizumab. So this bevacizumab is used for colorectal cancer, and there are more maps which are used for oral rectal cancer and each cancer. So these drugs will be used only for this uh, particular cancer. For example, if breast cancer is a patient is having breast cancer, they will be treated with trastuzumab. They will be given around 2 doses. So once these 2 doses, they will be checked if the patient's um, cancer, if there's any growth, or if the growth is stopped, or the growth is getting reduced. So that is the thing.
Another question is, sir, how the supernatant and pellet will itself be separated in the centrifuge?
Mm. So, yeah, can you show me that image, PPT, that centrifuge image? I think it is the third one.
Yes, sir.
Mm. So, ah, so yeah, you can see that top right corner, that is called as continuous centrifuge. In above that, there is a bowl. In a this thing. About the green motor part, there is a bowl. So inside that bowl, there are um, disc. Uh, there are discs which are upon each other, like 30 discs will be upon each other. So those discs will be continuously um, uh, rotating at the speed of 7,500 rpm. In 7,500 rpm. So from the top, you can see there are two pipelines which are going towards the bowl. So what will happen is, from upstream, the 2,500 liter of volume, it will be coming inside our bowl. So it will be continuously coming inside our bowl. So once this centrifuge, it is continuously running, it will, in that 7,500 rpm, it will separate the supernatant and pellet. So inside the bowl. So when it is getting separated, the pellet, it will be settled in the bowl's bottom, and the supernatant, it will be in the mid part, it will be separated. from supernatant to pellet. So the supernatant, it will get connected to the outer part, and to with the help of a motor, it will flow meter, it will come to the outlet. So the supernatant, once it is getting separated, it will continuously flow out of the bowl. This pellet, when we are having 2500 liter, our bowl's capacity, it will be around 70 liter only. But after, like for example, after 17 liter of flow inside the bowl, there will be a discharge of WFI, I mean water, AWFI, ambient water. So this ambient water, which helps to remove the pellet which is settled inside the bowl. So it will continuously beat it in a heavy force, so that the pellet, it will be discarded through the bottom of the bowl. So this process, it will be continuously happening for 17 liter once, so till the end of the 2500 liter intake. It will be continuously coming inside, separating, and it will be continuously going outside, and the pellet, it will be fleshing out each 17 liter once, till the end of the upstream's product intake.
Thank you so much, sir. I hope today all the participants must be benefited from your talk, those who are really interested in the downstreaming process. Next up, we have our second speaker, Mr. Shivakumar, who is a seasoned clinical data management professional with a strong foundation in end-to-end clinical data management, including trial setup, conduct, and close-out phases. With over 10 years of experience, Mr. Shivakumar has developed a unique blend of technical and interpersonal skills, including project leadership, data analysis, and collaboration with cross-functional teams. His expertise spans multiple clinical databases and tools, including Inform, Oracle Clinical, Data Management Workbench, and Power BI. Committed to delivering high quality results, meeting project deadlines, and exceeding stakeholder expectations, Mr. Shivakumar strives for excellence in all aspects of his work. We welcome you, sir. And today his topic will be in career opportunities in clinical research.
Yeah, thank you for the introduction, myself, Shiv Kumar, Senior Data Manager from Nordisk. Hope you are able to hear me.
Yes, sir, but it will be appreciable if your audio is much clearer, sir.
Just give me a second.
Yes, sir.
How about now? Is it clear?
Absolutely, sir.
Okay. Thank you very much. I'm myself, Shiv Kumar, and Senior Data Manager from Novo Nordisk. I'll be taking you to the career opportunities in clinical research. So before taking you to the career opportunities in clinical research, first of all, I would like to share my experience with respect to clinical research journey. Once I completed my bachelor's, and I thought it will be good if I take up the master's in biotechnology and completed in Karunia University MTech. Post that, it was not a cakewalk for me to get into the job. So initially, I just joined the diagnosis lab just for 1 month because I didn't want to waste even a single month after my master's. So I got a job in diagnostics labs. Post that, I got a BCIL internship, and this is also a good opportunity for the students who have completed the in academics, they can get an internship in a very good companies through BCIL. Once they write the exams, they'll be placed in internship for 6 months, which I got in Biocon. Post that, I went to the IQVIA. So I was introduced to the clinical research in the data management field. That was a very new 10 years back in so it was very new industry in India and Bangalore. It was developing, and companies were just coming to Bangalore from all over the globe 10 years back. So I joined IQV as a data. Are you able to hear me clearly, right?
Yes, sir.
Okay. Uh, So once I joined IQVIA as an internship, I completed a 3 months of internship and then completed 2 years there as a data manager. And then post that, I just moved to Novo Nordisk. So overall, as a data manager, I completed 11 years as of today. So with the 11 years of experience, I just want to give a glimpse of clinical research opportunities available where many people not be aware. There's a huge demand for this job opportunities within this, and there's a lot of domains. So who's interested in programming or other areas. It's not only the IT industries or wet lab or research where you need to pursue a career. So this is a very good opportunity to serve the patients who are waiting for new drugs or molecules. Waiting, the millions of patients are waiting across the globe. So it's also a wonderful opportunity to serve the patients. So with that, I will take you through next opportunities. Yeah, next one. Thank you. So here it's a clinical research industry. I would like to give a small background, like how it is currently. So it's a booming industry which offers rewarding career options to the aspirants. Also, it plays a vital role in introducing new drugs and improving health statistics. So if some patients are taking any insulin or for diabetes or any other diseases, maybe like a daily or twice a day. So if something comes like a weekly or monthly ones, like how good they feel. So that's how the research and the innovation has to be done. So here it provides the opportunity for innovation. And in India, the scope of clinical research is raising its global standards to introduce new medicines to cure various illness. And also in the upcoming years, India is going to become a global hub for clinical research. So as of now, from past 10 to 15 years, most of the global companies have come to India, especially the major in Bangalore, Chennai, Hyderabad, and Cochin. So all these places, there are a lot of companies, including pharma and CROs, have been established here, and there were like only 10 to 20 people joined 10 years back. Now there are thousands of people working in these companies. Yeah, next slide, please. And also the clinical trials market worldwide is worth around 26 billion US dollar, and the industry has employed an estimated of around 2 lakh 10,000 people in the US alone and over 70,000 in UK. So, and that forms a one third of total research and development staff. There are more than lakhs and 2,50,000 positions and maybe more than this vacant globally. This is just an estimation. A career in clinical research offers excellent job opportunities across the globe, including USA, Europe, Singapore, India. So now India is growing with this because most of the MNCs are moving their base to Bangalore and South India especially. India is going to be the hub in the upcoming future years. So, like for this, what is the eligibility? So, people will be thinking students like one who holds a bachelor degree in science can carve a niche in the fields related to clinical research. After graduation, one can explore the career in field having subjects such as life science, medicine, bioscience, and pharmacy. So, any bachelor's is sufficient, but masters will be good to pursue higher in the career ladder. Especially in this industry, for pharma industry, they prefer 2 to 3 years of experience. Then you might ask, like, how would the freshers be able to join? So there are some companies and CROs, they'll be hiring freshers, but it's not many like IT industry, they'll just go and hire from a campus. So it's a very small industry. So I think people just need to get training outside or explore the job opportunities available and apply for it as a fresher wherever it's available. And some of the required skills for this enter into this field would be like clinical research. One would develop so public apart from the knowledge on the clinical research, you should also have a public dealing how to because you once you enter into this field, we may have to um, engage with the people across the globe, and also capability to extract work from many stakeholders, and you need to have soft skills, very good communication skills, and capability to adapt to various cultures, because when you interact with different country people, so you need to know like how their culture will be, and like how do we need to get adapted to when we are working from different countries. So usually we work with 10 to 15 countries on a daily basis. So when we interact, we need to know the cultures and differences, and a keen observer and a keen eye for details. So it means like apart from what career, clinical research background, what you'll be taking as a skill, you should also have a good soft skills and a good spoken English and communication skills. Yeah, next slide, please. So opportunities by profession. So once you might be wondering like what are the job roles that are available within the clinical research. So here are the multiple roles and designations that are available. One might not be aware of it. Starting from study coordinators, I'll be taking you to. So once the clinical research will be done in the hospitals, especially in India. So one example I can tell, some trials will be conducted in CMC Vellore. And so there, like how the data, when the patients comes into the um, trial and clinical research, when they collect the data, so that data will be entered into the database by the study coordinators. So starting with that job role, so once the data is entered, so next moving into the clinical research associate, the second position, where they'll be interacting with the CRO companies or the pharma industry along with the investigator. They'll be acting as a bridge between the site staff with the hospitals and the company. So they'll be coordinating in getting the data and also validating source data. So, and the next will be the clinical research manager. So they'll be based in companies with the CROs on the pharma company. So these people will be developing the study and also preparing the protocol and also preparing the budget and coordinating to as a, who in charge of the whole clinical trial to from the start till the end. And next will be the business development managers, especially related to the CRO companies, where they need to develop their business and interact and get the more clinical trials from other pharma industries. And the clinical data manager. So where I'm also working as a clinical data manager. So here we play a crucial role in collecting the data once from the site. Once it's updated into the database, we ensure that data is up to date and cleaned because sometimes the data is not up to the mark as per the industry standards. So we collect all the data from all the countries, and we ensure to submit the data to authorities for the approval of the drug. So this we play a crucial role in maintaining the database and submitting. And drug safety associate. So once the collect the data has been collected in the database, so there will be a lot of adverse events and serious adverse events where patient may have to be hospitalized due to adverse events. So maybe may or may not be related to the drugs. So safety person will be involved in analyzing the effects of that adverse events. So that's also one of the crucial role in this industry. And database developments or programmers. So to collect the data, there are also standard database like Oracle, Inform, Rave, and others. But also within pharma companies and CRO, we would require database programmers to develop the internal systems and update or for the internal requirements. So that's also very good. Who are interested in programming, they can choose the database programming as a profession. And next we have a HTTM programmers. Once we collect the data, so that data has to be standardized in a steady data tabulation model. So that's a standard module where data will be submitted to the authorities across the industry because HTTM has come up with one single data HTTM model. So where because each company might have collected data and submitted in a different format, but the organization has come up with to develop and combine in one format so that it will be easy for authorities to analyze the data across the industry. So HTTM programmers will be looking into the HTTM data and collecting that. And next coming to the ones data is generated in HTTM. So next going to statistical programmers. So statistical programmers and statisticians, this is a very good role where they analyze the data, and also statistical programmers develop the programs, the data in a required format for in Adam programming to submit the data. So statistical programmers also, it's if someone is interested in programming and knows SAS, they can pursue career in statistical programming or as a statistician where there's a lot of scope in the industry. And next comes the medical writer. Once all the data is ready, so medical writer has to write uh, the report to be submitted to the authorities. Single report. So medical writer plays a crucial role to prepare the report of the complete clinical trial. And the regulatory affairs. So regulatory people will take care of all the regulatory matters involved in getting the approval. And then comes the auditors and quality assurance. So auditors also is important because they are within the company and within all these departments, and also to check the audit and inspections within the site, like everything is being performing as requirement or as per protocol. The auditors and quality assurance is also a department within this industry. So these are all the roles available where anyone can choose any of the interested career path in these roles. So these are the opportunities available. Okay, so some might be thinking like what might be the remuneration at the initial. So within this clinical research industry, starting for a fresher, it will be less than compared to the IIT field. It will be like around 10 to 20,000 approximately. But as and when you progress within the career ladder within the 5 to 6 years, it will be almost equal to the IT professional. So that's what. But later stage, I think it will be even good in this industry, especially you need not do higher studies as like PhD and postdoc. So within masters itself, you can go up to the director and also VP level if you are skilled enough. Okay, next slide, please. And futures perspective, India is the second largest pharmaceutical country in the Asia, and there seems to be a rise approximately 10% of the career prospects annually in upcoming years. So when I joined, there were like few tens to 100 employees working in each company. Now there are almost thousands of employees based in Bangalore for in each company. So you can imagine like what's growth of the industry within this. And various multinational companies are offering their research facilities in India, which result in exponential rise in clinical research industry. So as per study, the expected rise will be more than 50,000 in career options related to clinical industry. So after completing graduation, one can get a job at entry-level positions. For higher post, a master degree might be required, and somewhere as a specific certification courses are also required. So those who are willing to do a deep research, if you want to do a clinic completely into the research field within this, maybe PhD is also ideal solution. So, and along with the education fulfillment, a good hands-on is a mandatory to be in this field. One can start his career as a clinical research associate or as a fresher, followed by a project manager, and pursue like if they're skilled enough, can go up to director on vice president level. So with hard work, practical knowledge, and effective communication skills, can lead one towards the edge of clinical research industry. And here are some of the companies that I would like to mention. This is a very limited number I've taken available right now and where they're hiring. But there are many more companies across the globe where all the roles what I mentioned, the people being hired. So some like famous one like I want to mention it, like IQVIA, Accenture, LabCorp, Icon, PPD, Fortria, and TCS, Novo Nordisk. So all these companies you can just explore in this. So this was just a short introduction about clinical research. So if you have any questions to any of, you can just post it.
I think in chats they have mentioned, sir. How is AI beneficial as specialization with biotechnology? Which one is better if it's genomic engineering, drug engineering, or AI as specialization with biotechnology? They want to know which one is better: genomic engineering, drug engineering, or AI specialization with biotechnology.
As of now, actually, we can't tell which one is better because each one has um, uh, its own good. Because a genomic, if you wanted to develop a drug during the preclinical research, because starting phase zero, there will be like hundreds of molecules which will be developed. So genomic engineering is that time it will be helpful to choose the drug. But AI will be helping throughout from phase zero to phase four throughout the clinical research, including the genomic and also during the data management, study conduct, and analyzing the data. So AI will be helpful throughout, but only AI will not help because AI is still in a starting phase. Okay, so not much development has been seen in AI in the current industry. So it needs still a lot of improvement in the AI going forward. But I think definitely that will be helpful.
Okay. Then they are asking whether MTech in computer is advisable for this, or any specialization course is required for clinical data analyst.
I think life science background will be the preferred one. But if you're a computer engineer, they only be selected for the programming part. But if the life science background, they can get into any of the fields, and after like few years of experience, they can move within the industry, from HTML programmer to data management, or any role they can move within.
Then they want to know whether drug engineering is a good option.
Yeah, drug engineering is a good option, but in India, I think the opportunities are less, I feel.
Then they want to know which biotechnology field is like fastly developing right now, which can be like opt for further futures.
Sorry, can you repeat the question?
Which biotech field right now is developing fast?
Which biotech field?
Yes, sub field of biotechnology. That I think that's what they want to ask.
Mm,
Question says.
I think subfield, I can't answer that question particularly, but that complete clinical research is developing as such. I can tell all the fields are developing. So because of biotechnology, as such, has a very few opportunities, but biotechnology and clinical research combined has a lot of opportunities. But if anyone has pursued a career in biotechnology, can get into this clinical research or into the wet lab. So they have both options.
Okay, sir. Then they are asking which country like to in reference to your previous question, which is which country is good for drug engineering, like which country has good to scope for drug engineering course.
That I'm not exactly sure.
Then they are asking, is master required for clinical research to
Mm,
get into a higher position?
Yeah, that would be preferable. But even bachelors also, we have seen some have moved to the higher position based on the skills. But masters, if they have done, that will be a preferable. It will be good for their career ladder once after maybe 10, 15 years.
Then they are asking, can they complete medical coding and become a medical writer?
Yes, they can do it.
And then they are asking what are the procedure of the join to join the companies which you have mentioned in your PPT.
First one is you need to see uh, in the available for a fresher. So if they're a fresher, they need to see like they'll be posting some of the companies as in they'll be taking as an interns. They will not give directly the job. So first interns they need to join. They need to give interview for the interns as well. And if the candidate is good enough to pursue the career, they will hire that person after 6 months. Uh, or they can also take some courses outside in the private institutes if they offer campus interview or they guarantee the job. For example, some of the SAS institutes they have tie up with some companies, and if they complete the base SAS and advanced SAS, they will be getting into the directly into the companies once they complete the course. So they can take those courses and get into the companies only if they have types, or else they'll be freshers and internships. They can get into internship. That's one of the best opportunity. But usually, pharma companies will not prefer interns. They especially prefer like 2 to 3 years of experience. So that's one of the drawback.
Okay, sir. Then they want to know which country is good for MSc biotechnology. Is there any country which is
MS biotechnology, like there are many European and US countries. In Europe, where like Germany will be good, and also France, Germany, and US also will be good.
Then they want to know how to find internship in bio industry. Like, is there any website or any place that they can look into actively?
I think they'll be posting advertisement or once the opportunities are available or positions are open, they'll be posting in LinkedIn or somewhere they need to keep searching for that. There's no particular website. Maybe they need to go into the each company website and then see for job opportunities. If they're hiring for a fresher, they can apply for that. Or also if they have some contacts or some seniors working in those industry, they'll be aware of all the opportunities so they can get through the seniors or some through contacts.
Thank you, sir. Is there any more questions? Is there any opportunity for biochemistry in clinical research industry?
Yes, any life science background can get into clinical research.
Then they want to ask which elective course would be fit for this clinical research field.
There is no specification actually because for a fresher opening, there there'll be like lot of opportunities. Okay, any course they pursue, whether biotechnology or MSc biochemistry or whatever it is, so they don't take based on that. So during the interview, if the candidate is skilled enough, that's the only thing they consider.
Then they want to know whether BSc pass out can join to these companies.
Yes, yes.
Are there any special requirement, like it's just BSc, so like do they
No,
need
there is
any?
no any special requirement. The only thing is they need to find openings for the freshers. That's it, and they need to clear the interviews. There's no special requirement because even I have not taken any special courses or anything. Just I took the interview, cleared it, and then once you clear the interview and get into the company, all the training will be provided by the company for you to perform the job.
Are there any more questions? So they want to know what usually they ask in interview, like during placement drive or anything, what they ask in interview? The common questions which they should
Okay,
know
for
about.
common questions, like they should be aware of the clinical research from phase 0 to phase 4 and the regulatory requirements, and like how the clinical research will be conducted, and what is the scope of it. So phases of clinical trials. So there will be the basic questions about the technical interview, and also there will be a soft skills interview related to soft skills. So they'll be testing on that, and also aptitude test will be there. So it'll be different technical aptitude and also soft skills. Especially they care about the soft skill more than technical, because people will be involved in communicating or interacting with multiple people across the globe. So they need to be able to handle all the people and take the decisions and to communicate and get the work done.
Are there any more questions? So they want to know whether Biocon offers any biotech training course or internships.
Biocon will not offer any course as such, but they provide an internship. So every 6 months, I think they provide a internship for a lot of students. So I think they can easily get into the Biocon for internship for 6 months, or through the BCIL also. Once this is a Biotech Consortium India Limited. Once you apply for there, once you write an exam and clear that, so that's also an opportunity to get into. You'll also be getting a stipend of around some 10 to 12,000 approximately.
Uh, they also want to know, are Amazon or Google hiring biotechnologists? If yes, then what are the roles they offer to biotechnologists?
Amazon and Google, I'm not sure about it, because still in India, I think where clinical research, I'm not exactly sure if they have started clinical research by Amazon and Google.
Are there any other IT companies which offer these roles in India?
IT companies, there are a few like TCS I mentioned, right? So there are very few companies, but CROs, there are many CROs and pharma companies.
Are there any more questions? Devashi wants to know, sir, that someone suggested her to study a particular field in your MSc Biotechnology. Is it important to do MSc Biotechnology? I'm quite not understanding your question. Devashi, you can unmute yourself and ask the question.
Ma'am, I can tell,
Yes, yes, please.
ma'am, in future, or I can study in particular field in biotechnology, or can I study in continuously in MSc biotechnology? Important particular field is important to study.
I think it depends on your scope of interest. Like if you want to pursue your career within specific field, for example, microbiology or any research or stem cell research, then you may have to choose the specialization. Otherwise, to get into career, I think any background is sufficient, because when they take for an interview into the company, they don't see like what specialization exactly you've done, because the industry doesn't require what you have learned in the career. Most of the things will be taught by the company itself when you get into the company.
So there is one more question. They are asking
Thank you, sir.
Yeah.
if there is any entrance exam for this field to get job in this field.
No, there is no common entrance because the industry as such is a very small industry, and we should keep searching for jobs. But if you have a few years of experience, then there is a lot of scope in many countries. There's a lot of job opportunities available. Just you need to ensure you complete like 1 to 2 years as a fresher. Then you can move into any field within the clinical research and also across the globe.
Are there any more questions? Any more questions? If not, then we'll end the session over here. I would like to express my sincere gratitude towards Shivakumar sir for giving his insightful contributions and sharing his knowledge with us today. Your expertise will guide us through many things. Thank you so much, sir, for your time today.
Thank you. Thank you for providing this opportunity.
And for the participants, shortly I'll be sharing the link for feedback for your attendance in the group. You may leave the meeting. Thank you, everyone.